Hello, welcome back. For this next mini lesson, we're going to finish up the growth requirements, like molecular and chemical requirements, but then I'm going to also tag on to the end of this the ecological associations. So I'm going to jump over physical requirements because each one of these, just the nitrogen requirements and ecological associations, they didn't need to have their own video, so I'm going to squish them together and then do one on physical requirements next. Okay, so we just got done talking about chemical and energy requirements and oxygen requirements. So now we're going to take a look at nitrogen and some other like trace minerals. So nitrogen is needed in our macromolecules of proteins and nucleic acids, so like our DNA and RNA. So that's really, really important because if we didn't have proteins, we wouldn't have enzymes or structural proteins or pretty much anything. So nitrogen is really, really important. Now, in our atmosphere, we have about 80% nitrogen gas and about 20% oxygen gas. Now, that's most of our atmosphere is nitrogen gas. That's great. However, we as living organisms, most living organisms, cannot directly use N2. We cannot use nitrogen gas. So even though we have a ton of atmospheric nitrogen, it's inert to us. We can't put it in our molecules. We can't take nitrogen gas and make it into an amino acid or nucleotide, a nitrogenous base. But luckily, there are some organisms that can. And these are called nitrogen-fixing bacteria, and they typically live in soil, usually in association with other organisms, mainly plants. So if you remember back to chapter 11, we talked about the Rhizobium bacteria. They lived in those little nodules that stuck to the bottom of those legume plants. So their job, and they're really good at it, is to convert nitrogen gas into usable forms of nitrogen like nitrites or nitrates, and ammonia. So basically it's taking this N2 gas, which is an inorganic substance, hooking it together with some carbon and oxygen, which then maybe some hydrogen, which then is a form that allows our cells to use it. So these nitrogen fixing bacteria fix it into the plants use it, the plants now have these nitrates, nitrites, and ammonias in their tissues, and then we eat the plants, which then give us those usable forms of nitrogen. Or the animals eat the plants and then we eat the animals, food chain, food web, circle of life type of thing. And so then this usable form of nitrogen just kind of is now part of our ecosystem. This is called the nitrogen cycle that we're seeing in this picture here. So then plants die, animals poop, animals die. And so the nitrogen goes back into the soil as nitrates, which can then get recycled and used again in other plants. However, there are some processes that's called denitrification, because if this was only a one-way path, we would soon suck out of the nitrogen all out of the atmosphere. But we are in a pretty steady equilibrium with nitrogen in our atmosphere. So denitrification is breaking down the nitrates, nitrites, and ammonias back into N2 gas. So it's a cycle, right? So this denitrification is the opposite of nitrogen fixing. And there's some bacteria that do the denitrification as well. Sometimes lightning in a storm can also denitrify the nitrates, nitrites, and ammonias. So the nitrogen a lot of times is a limiting factor for growth, because if you don't have enough of these usable forms of nitrogen, you're not going to be able to grow. Your growth is limited. So this is our nitrogen, Rhizobium, essential for life on Earth. So I don't have a slide of it, but just going through the notes, the other essential requirements besides your four major atoms, carbon, hydrogen, oxygen, nitrogen, right, are hunk. Phosphorus, sulfur, calcium, magnesium, copper, iron, and a few other elements are only needed in about 5% of all of our compounds. So these are what we call trace elements. They just are needed in really, really, really small amounts. You might also need growth factors, which are not atomic. They're not atoms or elements, but they are a combination of those things. So maybe like vitamins or cholesterol, fats, and lipids, NADH, NAD+, these electron carriers. So those are also considered growth factors that are needed in relatively small amounts. All right, so those are kind of the other. So we did energy, carbon, oxygen, nitrogen, and other. So those are those types of requirements. Now I'm going to jump ahead in the notes. We'll skip over physical requirements. That will be the next video, and jump to ecological associations, because that's just a short little section I'll tag on to the end of this. So you may have heard of these terms before, like symbiosis, mutualism, commensalism, those types of things, parasitism. We're going to be on the higher scale of this, because we're going to get to those topics again in chapter 14 towards the end. But we're going to stay with just large scale topics like antagonistic relationships and synergistic relationships. So antagonistic relationships are where one organism harms another. Synergistic relationships are where they work together to be benefiting. And symbioses can fit into the synergistic or antagonistic, depending on which symbiotic relationship you're taking a look at. One of the things, though, I wanted to talk about were biofilms, because in the bacterial world, biofilms are a really important ecological association, not only for the bacteria themselves, but for humans, because biofilms increase the pathogenicity of certain organisms. So what is a biofilm? So let's say you have a surface, right? Oh, let's get an actual color you can see. So we have a nice flat surface, and a few bacteria come around and want to stick to it. So they might have a capsule or a slime layer, or some fimbriae, and they just start sticking. And when they get close together, they communicate. They have these, what's called quorum sensing. So quorum sensing right here. These quorum sensing molecules that allow them to talk to each other. And once they do that, by recognizing that other bacteria are sticking in the same place, they start turning on genes, probably via an operon, that they wouldn't have if they were all by themselves. And so now they start showing different phenotypic characteristics that makes it easier for them to stick and be part of this association. And so it changes their biochemistry, it changes their shape, it changes the genes that are expressing, and then more cells come in and you get this big blob of bacteria and extracellular goo. Some water can move through it, but these cells are behaving differently than if they were all by themselves. And a lot of times, some of those genes that they turn on when they're in this big group are genes that make you sick, these virulence genes. I'm going to post a video in the helpful links for a wonderful TED Talk that talks about this little squid here, this cute little squid, and a beautiful symbiotic relationship it has with this bacteria that bioluminesces. And she talks, the woman's name is Bonnie Basler, and she talks about quorum sensing and how the squid take advantage of this quorum sensing between this Vibrio fischeri, and how humans are trying to take advantage of this communication between microbes to maybe help with antibiotic resistant infections. I always play this video in class, so I strongly encourage you to also watch it when you're studying this chapter 6 stuff. It's really amazing. All right, so that's all I wanted to talk about with this little mini lesson. So we will get on to physical requirements in the next video. See you next time.